Bonds are interactions between atoms. Right? So, atoms have electrons outside of the nucleus. The outermost electrons are the valence electrons, and those valence electrons interact with other valence electrons from other atoms, and that interaction is what we call as chemical bonds. And chemical bonds, we saw there are many types of chemical bonds, many types of interactions between atoms, and the more important ones for biology are covalent bonds and ionic bonds. Ionic bonds include giving and taking of electrons, whereas covalent bonds involve sharing of electrons. When the sharing of electrons is equal between the two atoms, then there is no charge distribution between those two atoms, in which case it is called as non-polar. An example of this is the methyl group. Carbon and hydrogen have the same level of electronegativity. Electronegativity is the ability of atoms to attract electrons towards themselves. So, because carbon and hydrogen have similar electronegativities, their attractive forces on the electrons are equal, and therefore neither carbon nor hydrogen has any charge associated with them. But on the other end, for example, with the hydroxyl group, when you have oxygen and hydrogen sharing electrons, oxygen has a much higher electronegativity compared to hydrogen, meaning that it has a much greater capacity to pull electrons towards itself. When it does that, because the electrons are closer to oxygen than they are to hydrogen, electrons are negatively charged, right? So if the negatively charged electrons are closer to the oxygen, then that means that the oxygen is going to be partially negatively charged, and the hydrogen is going to be partially positively charged. So this is a polar molecule because the charges are distributed at the poles. An example of a polar molecule is water. Water has an oxygen atom and two hydrogen atoms. And as we just discussed, the electrons are going to be pulled closer to the oxygen, in which case you are going to have a slight negative charge represented as this delta, delta negative, delta minus. So you're going to have a slight negative charge here, and the hydrogens, which have kind of lost their electrons in some way to the oxygen, are going to get slightly positive charges. So you're going to have delta plus, delta plus. When you have a lot of water molecules in a system, the polarities of these molecules are going to play a role in how these water molecules are going to interact with each other. So, for example, in this case, you have an oxygen with its partial negative charge being attracted to the partial positive charge of the hydrogen of another water molecule. So that attraction is what we call as a hydrogen bond. Okay? So this is hydrogen bonding. This hydrogen bonding property, becomes the key to a lot of the properties of water that we know of, which becomes important for life. One of them is cohesion. Cohesion is the interaction of water molecules with other water molecules. Adhesion is the interaction of water molecules with other substances. For example, when we look at how water is transported during transpiration, which is that when how water is moved from the roots to the leaves, where water is lost through evaporation, what happens in this process is water is transported through a narrow tube called as xylem, through many narrow tubes called xylem. And xylem, the inside portion of xylem, has polar properties, has polar molecules. Water being a polar molecule, it can interact with the inner surface of that xylem, and it starts because of this upward suction happening because of the vacuum created by evaporation. These water molecules are going to slowly glide along the inner surface of the xylem because of adhesion. So, but as the adhesion is moving the water molecules up, it's crawling up the surface like this, it is also because of cohesion pulling the other water molecules alongside. So adhesion and cohesion together work to create transpiration possible and making moving particles up the xylem. The other property that we just saw was surface tension. Surface tension is, so any surface again is a bunch of particles attracted to each other through chemical bonds. So when we have when we have water, the molecules at the top of the water surface are forming bonds with all the other molecules around them, whatever molecules they can around them. And that's creating a little surface with water. So that tension that is caused is called a surface tension. Another property of water is high specific heat. Specific heat capacity is the ability of, is the amount of heat that you need to move or increase the temperature of a system by a degree. So what that means is water, because of its multiple hydrogen bonds, is going to require a lot of heat applied to it so that it can break all those hydrogen bonds and become free. So that is why water has a very high specific heat. In the same way, water can exist in three different forms, three different phases. One is as a solid, and one is as a liquid, and the other one is as a gas. Gas is water vapor. So in these phases, you need to apply a lot of energy to move from one phase to the next phase, from being solid to becoming liquid, and from being liquid to becoming gas. It's going to take a lot of energy. The energy required for phase change is called as latent heat of vaporization, and water has a very high latent heat of vaporization because of the number of hydrogen bonds that it has. Water is less dense than ice. We just saw the picture and we went through that exercise, so you're familiar with that. And water is also a universal solvent. What that means is it is capable of breaking ionic bonds. So for example, if I have sodium chloride is an ionic molecule, is an ionic compound. And it's because sodium is willing to give one electron to chlorine, which needs one electron in its outermost shell. So what happens is when sodium gives away an electron, it becomes positively charged. And chlorine, because it receives an electron, becomes negatively charged. Now imagine this being in the presence of water. Water, because of its partial positive charge, the oxygen is going to get attracted to the sodium. And it's going to surround the sodium. Same way, other water molecules, the hydrogen with their partial positive charge are going to get attracted to the chlorine, and they will surround the chlorine. This is what we call as forming a solution, dissolving of a substance. And a lot of materials in nature are ionic compounds. Therefore, the presence of water causes them to break apart. This is why water is called as a universal solvent. Yes, sir?
So hydrogen surrounds the sodium and then...
No, water surrounds the ions.
Okay, and then it attracts the atoms and the ions and then dissolves them? Or how does it...
Sorry,
So does it like attract the atoms in table salt and then break it apart and dissolve it? How does it work?
Yeah, if you have an ionic compound, right? So when you have an ionic compound, what it is you have sodium and chlorine, now with opposite charges, right? Now the opposite charges, positive and negative, are going to attract each other. That bond is called as an ionic bond. And when you have water, the forces of binding of these multiple oxygens to the sodium is greater than the binding force between sodium and between these two ions. Therefore, this breaks apart and this breaks apart.
Okay.
That's what dissolving something is. Okay, and then we want to look at the elements of life. That was chapter 2. There are, even though there are 118 elements in the periodic table, 6 of those are most relevant to life. They are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And these are specifically suited for life because of their ability to form covalent bonds, and also because they are at the right size. If they are too big, then the kind of bonds that they form are not very strong. For example, when we looked at phosphorus and arsenic have the same number of valence electrons. But when you and do you remember where you find phosphate groups?
Nucleic acid.
Nucleic acids, backbones have phosphate groups, right? So imagine arsenic replacing phosphorus. It will still be able to form the same kind of bonds that phosphorus forms. However, what happens is that arsenic is not able to hold on to the electrons to the bonds that it has formed. So because of that, it will break as soon as it forms. So imagine replacing all the phosphorus in that in the backbone of your DNA with arsenic. It's going to form the same kind of bonds. It's going to form an arsenic sugar backbone, but soon afterwards it's going to break, which means your DNA is going to break, which means your organism is going to die, which is what we call as arsenic poisoning. So, of these 6 elements, carbon is particularly suited for all of life. In fact, all of life on Earth as it exists today is built around carbon. So, carbon can form 4 bonds because it has 4 valence electrons, and those bonds can be single bonds, double bonds, or triple bonds. They can form a lot of combinations with other atoms, and they can form 3-dimensional structures because of the angles of those different bonds. It can form 3-dimensional structures, and 3-dimensional structures are important for biological functions. Complex structures are important for biological functions. So, looking at the 4 major biomolecules, we have carbohydrates, lipids, nucleic acids, and proteins. They have specific combinations of these atoms. Carbohydrates have carbon, hydrogen, and oxygen in the ratio of 1:2:1, and lipids have carbon, hydrogen, and oxygen, but the amount of oxygen is much lesser compared to oxygen. Compared to carbohydrates. Proteins have carbon, hydrogen, oxygen, nitrogen, and sulfur, and amino acids, nucleic acids have carbon, hydrogen, oxygen, nitrogen, and phosphorus. Yes, sir.
Do we need to memorize what has what? Like-
Yes, absolutely. You need to know which has what. So the question is going to be something like this. Questions could be something like this. Scientist A worked on synthesizing a compound, and in the compound he discovered that he isolated had a composition of carbon, hydrogen, oxygen, nitrogen, and sulfur, but it did not have any phosphorus at all. What kind of a compound has scientist A created? Yeah. So he has created a protein, right? He has created a. So that's the kind of information that you need to be aware of. Yes, sir.
And then you need to memorize the ratios too, right?
Don't need to.
Okay.
What you probably need to know is, let's say, scientist B created a compound which had carbon, hydrogen, and oxygen in the ratio of 1:2:1. However, but he also made synthesized another compound, but this compound also had carbon, hydrogen, and oxygen, but the ratio of oxygen seemed to be much lesser than compound 1. So what would that compound be? Lipids. So stuff like that. Then we looked at functional groups. We saw there are functional groups, or groups of atoms that have a unique function, that have a specific function. And there were some functional groups that are especially relevant in biology, that includes carboxyl groups, COOH, carboxylic acid groups, amino groups, NH2, hydroxyl groups, OH, methyl groups, CH3. So what's the nature of methyl group? Non-polar, yes. Methyl groups are non-polar. Carbon and hydrogen, we just discussed, they share share electrons equally, right? And then phosphate groups and sulfidyl groups. Where did we see sulfidyl groups in the last class? We saw sulfidyl groups before. Anyone? Something about proteins, protein folding?
Oh, the bonds between that creates the folding of proteins.
Yes. So there's an amino acid called cysteine which has the sulfidyl group. When you have 2 sulfidyl groups coming together, they form a disulfide link, disulfide bridge. So the sulfur and the sulfur can form a covalent linkage called disulfide link, which helps shape the structure of a protein. Okay, and then Naisha, you had a question? Yes. So functional groups is one of those things you have to memorize. You need to know which is what, and some basic properties about them. Like COOH is an acidic group because it's going to give out hydrogen. Ammonia, the amino group is going to receive hydrogen, so it is going to be positively charged. So those are the basic things you need to know.
Right, so that's properties, but do we need to calibrate the stuff as well? Like the structure?
Like CO and all,
Yeah,
you don't need to know that.
Okay.
Yeah. Yes, ma'am. You had a question?
Oh, no, sorry.
No. Okay.
So on the slides, does it? I can't remember. Does it say which are non-polar and polar and things like that?
It does.
Okay.
Arsha, you had a question? No? Okay. Alright, so The other thing we looked at very briefly was isomers. What you need to know about isomers is that isomers have the same chemical composition, but they have different organization of organization, arrangement, and positioning of atoms. So, for example, we looked at glucose. Do you remember the two forms of glucose we looked at? Glucose. We saw the ring structure of glucose. And then we said there's a hydroxyl group that either points up or points down.
Oh,
Beta,
beta,
alpha.
Alpha and beta, right? So if you look at the chemical composition of those two, it's the same. It has the same amount of hydrogen, oxygen, carbon. Yes.
Could you repeat your definition about isomers?
Isomers have the same chemical composition, but they may vary in the arrangement and positioning of atoms. So, that is a type of isomer, okay? Alpha and beta isomers of glucose. So here I have drawn a picture where you see you have carbon, 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 with the hydroxyl groups on one side, hydrogen on one side. What I have done here is I have shifted the hydroxyl group on first carbon to this side. So these two are isomers. Okay. But one of the isomers that you need to remember, and that you will probably encounter frequently, is the idea of cis versus trans isomers. So let's say there is a carbon atom bound to another carbon atom, and this carbon atom is bound to something called X. I don't know, I just made up something. And this carbon is also bound to X. If the X's are on the same side of the bond, they are facing, both of them are facing up, then you have a cis isomer. When you have the X's facing the opposite directions, one is up and one is down, that's a trans isomer. Okay? So you need to know cis versus trans isomer. You might encounter this word enantiomers. Enantiomers are so they are mirror images of each other. Think of your hand. Hands are alike, but you can't superimpose on them because they are mirror images of each other, right? So they are called enantiomers. There's an L enantiomer and there is an R enantiomer. Yes.
So for the alpha and beta isomers, which is like the one with the all the hydroxyls on one side, and which is the one with one of the hydroxyls swapped with the hydrogen?
So the difference between alpha and beta glucose, we're talking about glucose, right?
Yes.
Yeah. So the difference between alpha and beta glucose is in carbon number 1, the hydroxyl group is facing downward in alpha glucose, and the hydroxyl group is facing upward in beta glucose.
Okay.
So then we look at macromolecules. We have 4 types of macromolecules: carbohydrates. Yes, go ahead. Yeah. Video still on? Oh, video still on?
Yeah.
Okay. So there are 4 types of macromolecules. We have carbohydrates, lipids, proteins, and nucleic acids. Of these, carbohydrates, proteins, and nucleic acids are considered as polymers. Lipids not so much, because these 3 molecules are made of macromolecules are made of repeating monomeric units. The repeating monomeric units of carbohydrates are monosaccharides. For proteins, the monomers are amino acids. For nucleic acids, then monomers are nucleotides. Monosaccharides are bound to each other by glycosidic bonds. Proteins are joined together by peptide bonds, and nucleic acids are joined together by phosphodiester bonds. Amino acids are, just to quickly look at the structure, amino acids have a central carbon. All amino acids have a central carbon. On one side they have an amino group, one side they have a carboxylic acid group, one side they have hydrogen, and then they have an R chain or a side chain. Okay, the side chain can be anything. It can be hydrogen, it can be methyl groups, it can be a hydroxyl group, it can be it can be a ring structure, it can be many things. So that's the general nature of these amino acids. And nucleic acids have 3 parts to them. I'm sorry, nucleotides have three parts to them. They have a phosphate group, they have a sugar, which can be a ribose or a deoxyribose, and then you have a nitrogenous base, which is rich in nitrogen. Lipids are, yes sir?
Wait, so out of those three parts, which one, the nitrogen base, is the one that changes between DNA and RNA? Or,
One of them is different. In DNA, you have a nitrogenous base called theme or thymine.
oh, yeah.
In RNA, instead of thymine, you have uracil. Thymine is methyl uracil, basically. It's the same thing, the methyl group on top.
Oh,
So, so for these three, the process that creates polymers is through loss of water, and that synthesis is called as dehydration synthesis. For breaking a polymer, which is called depolymerization, you add water, and that process is called as hydrolysis. So, dehydration synthesis and hydrolysis are the two reactions that make polymers and break polymers. Now, carbohydrates can be classified as simple and complex carbohydrates. Simple carbohydrates include monosaccharides, which are single molecules, single glucose molecules, like glucose or lactose or galactose, and then disaccharides like maltose and sucrose. Or you can have complex polysaccharides, which can be either oligosaccharides or polysaccharides. Oligosaccharides are between 3 and 10 3 and 10 sugars long, and then polysaccharides are more than 10 sugars. The word OSE always refers to a sugar. The word glyco also means sugar. So whenever you see the word glycolipid, it means sugar lipid. Glyco protein, it means sugar protein. A conventional way to represent a sugar is in a cyclic form. Okay? A cyclic form like this. This is called as a ring structure, Hawthorne ring structure. So in the ring structure, you have a naming system starting with 1 going all the way in the clockwise direction. Yes, sir?
do we need to know which ones are like monosaccharides, which ones are disaccharides, and which ones are?
They'll tell you. They'll tell you. You don't need to know.
Okay.
So here I have drawn a pentose. Pentose means 5 carbon sugar because it's penta and ose, 5 carbon sugar. You have 1, 2, 3, 4, and the 5th carbon is sticking out. And I have 2 variations, 2 different types of pentoses here. One of them has a hydroxyl group in the 2nd carbon, and another one has a hydrogen in the 2nd carbon. The one that has the hydroxyl group is called as a ribose. But because you have taken away an oxygen, you are deoxidizing it. So it's a deoxyribose. So this kind of sugar with the hydroxyl group is called an RNA. This kind of sugar without the oxygen and hydrogen instead is called as is present in DNA. We already saw this, beta glucose and alpha glucose. So an arrangement of these molecules, of these monosaccharides, depending on how they are arranged, whether they are straight, whether they are branched, can give rise to different functions for them. So in plants, their carbohydrates are arranged one after the other in the form of starch. Starch is the storage molecule for energy in plants. In animals, that storage molecule is glycogen. Okay, so starch in plants, glycogen in animals, they are the storage molecules of carbohydrates. They also have structural functions depending on how they are arranged. Again, you can create strong structures. You're welcome. So they form structures. Examples of these are cellulose. Cellulose are present in plants, and then you have in chitin, which is present in insect shells outside, the hard exoskeleton of insects, as well as the material that makes up the cell walls of fungi, mushrooms. Okay, that's also chitin. Alright, so proteins. Proteins are, we saw, strings of amino acids. So you have an amino acid here, COOH, and you have another amino acid here, and the NH2 group pointing here. The water is lost. So what I end up with is a CO and an NH. The carbon and the nitrogen are bound to each other. The carbon of here, not the central carbon, the carbon that comes from the carboxylic group of this one, and the nitrogen that comes from the amino group of the next amino acid, they are bound to each other through a bond. That bond is called as a peptide bond. So the properties of the amino acid depend upon what the R group is, the variable group is. If that variable group is polar or non-polar or acidic and basic, the amino acid also becomes polar, non-polar, acidic or basic. If I have a string of amino acids attached to each other in one long sequence, that sequence is called as a primary structure of a protein. But within that, so soon after that primary structure is formed, hydrogen bonding happens between nearby amino acids. Nearby amino acids can form shapes depending upon the hydrogen interaction between them. So they can form helices or they can form these beta plated sheets. So if you look at a string, you're going to a sequence, you're going to see local interactions called as secondary structures, and there are two of them, alpha helices or beta sheets, making them up. This is the secondary structure. But depending upon the R group, depending upon the properties of this R group, within that straight stretch, you are going to have interaction. So maybe an amino acid here, the R group of this amino acid here, and the R group of this amino acid here may be positive and negative polarity. What that means is it's going to be pulled together, right? It's going to be pulled towards each other. And that means that you're creating a three-dimensional structure. So you're creating, so it's going to be bound upon itself over and over depending on all kinds of bonding, ionic bonds and disulfide bonds and polar bonds and hydrophobic interactions and van der Waals forces. So all of these things are going to happen here. And all of that is going to create a shape for that one single peptide sequence. So a shape that is formed, a three-dimensional shape with one single peptide sequence is called as a tertiary structure. Okay, a tertiary structure. But you have, for functional protein, sometimes you need more than one subunit, one tertiary structure. So you have multiple subunits of proteins coming together to create a functional protein, which is called as a quaternary structure. Yes, sir?
So quaternary structure is a bunch of tertiary structures put together.
We call these as subunits. Each one is called as a subunit. So a bunch of subunits of proteins coming together makes a quaternary structure. Now here's an interesting thing for you to remember. We did the apple brownout experiment, right? What was, yes?
So like, does the protein folding like occur more because like of the double bond between like the carbon and nitrogen, or is it like the bond between, I think it's like the functional group of like this, the S and the S, like the different.
The disulfide bonds?
Yes.
The disulfide bond is one of the many forces that bring together a three-dimensional structure. So when you're looking at the single sequence, the sequence of amino acids is called the primary structure. The local attractions that give them the hydrogen bonds, that makes alpha helices and beta sheets, that's the secondary structure. The R groups, which are present in the sequence of amino acids, their interaction, which could be ionic or disulfide bridges or polar interactions or non-polar interactions, all of these are responsible for the tertiary structure. Okay. Alright, so when we did the apple brownout experiment, why did your apples brown? Oxidation. And what do you think caused them to be oxidized?
The oxygen.
Definitely, but what caused the oxygen to, what is it in the apple that helped the oxygen bind to the materials there, to the phenolic compounds there? Anybody? No? Enzymes. There are enzymes present on apples, right? The enzymes that are present in apples and pears and so on that actually are able to use the oxygen and fix it into the compounds over there. So this oxidizing reaction happens because of enzymes. Enzymes are usually proteins. There are sometimes ribozymes which are RNA, but enzymes are proteins that have catalytic functions, meaning that they can make a reaction go really, really fast. And they do that by breaking bonds or creating bonds. In this case, breaking bonds. So all proteins need a particular range of temperature and pH conditions to work efficiently. So if I change the pH, which is what you did, you added vinegar, some of you added lemon juice to the apples and saw that it wasn't turning color, right? It wasn't changing color. What do you think happened to the enzymes that were fixing it? What did the change in pH do?
It broke the bonds of what was making the apple or pear oxidize.
Exactly, they broke the bonds of the enzymes. In other words, they denatured the enzyme. So when every enzyme needs a range, a particular optimal temperature for function, an optimal pH for function. When you disrupt that, when you change the pH or when you change the temperature, then it breaks the chemical bonds that give the structural that gives the structure of the enzyme, which means once those bonds are formed, the structure is lost, which means the function is lost, which means your enzyme is no longer functional. Which is why by changing the pH, you denature your enzyme and made it non-functional. So some of you are asleep, but let me quickly, I don't know, 2:26. What time do we finish? 2:50. All right, I'll be done in about 10 minutes, and then you can have the rest of the class. Okay? But I just want to get through this because this is all important for your exam. Okay, so nucleic acids are made of nucleotides. Nucleotides have three parts: phosphate group, sugar, and we saw that it's different between RNA and DNA. DNA has deoxyribose sugar, RNA has ribose sugar, and then you have your nitrogenous base. RNA has 4 types of nitrogenous bases, which are A, U, G, and C. All unicorns go to church. DNA has 4 types of nitrogenous bases: A, T, G, and C. All apples in the tree, cars in the garage. Okay, so that's one of the differences between DNA and RNA, that the nucleotides are different, and ribose versus deoxyribose sugar. RNA is typically found as single-stranded structures. DNA is found as double-stranded structures. And also need to remember that A always binds to T and G always binds to C. In RNA, A always binds to U and G always binds to C. The bond that forms between nucleotides is called as a phosphodiester bond. So if you go to the video that I shared yesterday, did anybody get a chance to watch it? Nobody did? Okay. All right. So if you get so please take some time to watch it. Okay. It's because I'm explaining in detail what the sugar phosphate backbone is and what the phosphodiester bond is, how that is formed, and what the 5' end, what the 3' end is. So I've gone through this in quite a bit of detail. So it's not too long. It's only about 17 minutes long. But I think it's good information for you to. So nucleic acids, follow through with your with the video or other resources. And then lipids finally. Lipids are basically generally divided into fats and steroids. Fats are further divided into many groups, many special groups, but the most common of those is triglycerides. Triglycerides have a glycerol molecule, so think of the uppercase alphabet E, and then you have 3 fatty acids attached to them. Phosphoglycerides, on the other end, have 2 fatty acids and 1 phosphate group attached. Now, fatty acids are long chains of carbon and hydrogen, which makes them non-polar, which means they don't interact with water. And then you have an hydroxyl group attached at the end. In fact, it is the hydroxyl group that allows them to bind to the glycerol molecule over here. And so, if you have a fatty acid where there are no double bonds, you call it as saturated. And these saturated molecules tend to be long linear molecules, which means you can pack a lot of them into a small space. So in room temperature, they tend to be solids. Examples of these are butter and lard. Most animal lipids tend to be solid. But when you have at least one double bond, then it creates a kink, which means that they are not able to pack together tightly. And these unsaturated fatty acids, because they are not tightly packed, tend to be liquid at room temperature. Examples are oils that you get from plants, olive oil and avocado oil and whatever thing. So those are the main things. Steroids are ring structures. You have 4 rings. There are 3 of them. There are 6 rings and 1 of them. You don't need to know all that. But steroids are ring structures of carbon, hydrogen, oxygen, and nitrogen. And these ring structures play a very important role as precursors of other hormones. Hormones are these molecules that signal changes throughout your body, signal information throughout your body. That's it in a nutshell. Any questions? All right. If you have any questions, come by, we'll talk. Okay? So if you want to spend some time thinking about planning for your make sure you finish your PPC, guys. Your progress check. Unit 1, it's called Unit 1 progress check. Make sure you get at least a 50% in that before the class, so that I can grade you. Okay?
Don't want to repeat class. Don't want to repeat class.
It's only